All right, we're going to look at the blood portion of your notes now. So, where you have blood, we have some characteristics. Slightly viscous, 4.5 to 5.5 is the viscosity factor, which we'll talk about in just a minute, due to the cells and the plasma proteins. Blood consists of cells, cells, and most of that's going to be red blood cells. These are erythrocytes. Erythro means red, and cyte means cell. So you see the erythrocytes here, all these little red cells. And about 1% of white blood cells will be in there. And plasma. Plasma is the liquid portion, which they're sitting in. So it consists of cells and plasma, about 99% red blood cells and about 1% white blood cells. A pH 7.35 to 7.45, so that's going to be slightly alkaline. Arteries have a pH of about 7.4 and veins about 7.35. The salinity, 0.85 to 0.9% saline content. If you go into the hospital and you need a fluid volume increase, they'll put you on a bag of Ringer's or isotonic solution. It'll be a 0.9% saline. Males have about 5 to 6 liters of blood. Females, about 4 to 5 liters. Now that's on the average because in general, males tend to be larger than females, and that's why there's a difference there. Here is something called the hematocrit, or packed cell volume, PCV. It's the percentage of blood that is formed elements like cells. And you can see that there's a sample being taken. That could be a test tube, it could be a pint, it could be a thimble, it could be a capillary tube. You're going to get the same results. So they took some and put it in a test tube, spun it down, and when they did that, the red blood cells and white blood cells were slung to the bottom because they're more dense, and the plasma containing electrolytes and water and some other molecules are up at the top. So the liquid portion's at the top, the cells are at the bottom. The buffy coat is there, it has leukocytes in there, usually. Males, about 42% of a sample of cells is formed elements. About 42% is formed elements, cells. Females, around 38%. So that means formed elements in males, 42%, so that's roughly 58% plasma. And you see that kind of falls in these ranges here. If you look at the picture, plasma, 55% of whole blood, erythrocytes and leukocytes, roughly going to be around 45% of that whole blood. Water has a viscosity of 1, and blood is 4.5 to 5.5. So above there it says the greater the hematocrit, the greater the friction between cells. Okay, that means that the higher the hematocrit, the more cells that are in there, it's going to be more friction. So where it says water has a viscosity of 1, water is used as the standard for viscosity around the world. Blood is 4.5 to 5.5. This means that 4.5 to 5.5 times as much pressure is required to force whole blood through a given tube as is required for water. Okay, blood is about 4.5 to 5.5 times harder to force through a given tube than water would be. Here's a red blood cell here. So it says formed elements, red blood cells and white blood cells. Well, here's a red on this page. It says erythrocytes or red blood cells contain no nucleus. They lose their nucleus prior to their release from the red bone marrow. Their major function is to carry hemoglobin. That's a molecule, which in turn carries oxygen. So red blood cells are packets of hemoglobin, and the function of hemoglobin is, one of the functions, is to carry oxygen. Males, that's the first one, 14 to 16.5 grams of hemoglobin per 100 mils of blood. Okay. Females have 12 to 15 grams of hemoglobin per 100 mils of blood. Now, hemoglobin, besides carrying oxygen, is an excellent buffer. It can also carry hydrogen ions, also act as a buffer. If you look at the diameter of this cell, the books change every so often. This says 7.5 micrometers. Well, I have 7.7 in diameter in my book. Well, that's in the same general ballpark. They're biconcave, so they cave in on both sides. And you can see how this caves in and this side caves in, and it makes it less dense in the middle. So it appears to be slightly hollow, which is actually not hollow. It's just thicker, more dense around the periphery, and it's thinner in the middle because it is biconcave. It concaves on both sides. Concaves in on both sides. Carries oxygen and carbon dioxide attached to the hemoglobin. So hemoglobin can also carry, carbon dioxide. So let's look at that molecule. Here's a molecule of hemoglobin. It's made up of 4 polypeptides, and they have iron in the middle. So you see Fe is iron. Iron is required to form hemoglobin, so it functions. It's responsible for the ability to bind to oxygens. The number of red blood cells in males is around 5 million per cubic millimeter of blood, and females 4.5 million per cubic millimeter of blood. On a metric scale, like a 6-inch ruler, if it's got the metrics outlined, those little bitty marks on the metric side, if it was a cube, 1 millimeter by 1 millimeter by 1 millimeter, it would hold around 5 million red blood cells for the male and about 4.5 million for the female. Formation of blood cells is called hematopoiesis. I'm showing here formation of red blood cells. So hematopoiesis for blood cells is HEMATOPOIESIS. And what we have here, formation of red blood cells, is erythropoiesis. 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 Erythro is red, and poiesis has to do with the formation. And you see here there's a stem cell called a hemocytoblast, that makes these red blood cells. And you can also see the third picture from the end there, how the nucleus is lost prior to its release. In the embryo, red blood cells are made by the liver and the spleen, or just blood cells made by the liver and the spleen. In adult, blood cells are made by the bone marrow and lymphatic system. Bone marrow and lymphatic system in adults. Embryos is the liver and the spleen. Now the lifespan of a red blood cell is around 120 days. That's like three months. The spleen and the liver remove the old red blood cells from the system. Now the spleen and the liver are going to be monitoring the flexibility of the red and white blood cells as they pass through the screening areas of the liver and the spleen. If the cell's membrane is flexible enough, it's allowed another pass. If it's picked up that the cell membrane is becoming a little more firm or less flexible, it is removed from the system, phagocytosed, broken down, and the elements are recycled. This has to happen because these red blood cells in particular have to be able to squeeze down and go through capillaries, and so capillaries are going to squish them. They have to be able to be reshaped and move through those capillaries and then rebound when they come out. The hemoglobin is broken down by the liver into iron and bilirubin, which is oxidized to biliverdin. Bilirubin is one thing that gives your urine its golden color. Now, production. A low oxygen concentration causes the kidneys to produce erythropoietin, erythropoietin, a hormone into the blood. So low oxygen concentrations, the kidneys pick this up and release a hormone called erythropoietin into the blood. Erythropoietin causes that should be bone marrow, to increase production of red blood cells. And so it's going to stimulate the bone marrow to produce red blood cells. You have a blank, hypoxia, HYPOXIA, cellular oxygen deficiency, cells not receiving enough oxygen. And then below that, there's another blank, anemia, ANEMIA. The number of functional red blood cells and/or their hemoglobin level is low, therefore the blood has a low oxygen carrying capacity. So you could have plenty of red blood cells that just don't have enough hemoglobin, so you can't form the hemoglobin. Or you could have plenty of hemoglobin per cell, it's just that you don't have enough red blood cells in your bloodstream. So things that could affect that. A lack of iron, and usually when people are diagnosed as being anemic, one of the first things a doctor will do will put them on an iron supplement. A lack of certain amino acids for the protein globin, because hemoglobin, the heme is a pigment that makes the blood red, and the globin is the protein portion. Lack of vitamin B12, which helps in red blood cell formation by red bone marrow. Now I put in there, number one, B12 is obtained from liver and meat. An intrinsic factor is secreted by the mucosa of the stomach, which picks up the B12, which is the extrinsic factor. Both go to the liver to form anti-anemic factor. Anti-anemic factor goes to the bone marrow to stimulate cell production. So it's kind of a process that goes on there, but it eventually ends up in causing the bone marrow to produce more red blood cells. White blood cell formation. From the bone marrow, you have, those are abbreviations for some cells we'll talk about in a minute, neutrophil, eosinophil, and basophil. And lymphoid tissue is going to be lymphocytes and monocytes. And we'll look at those in just a minute. So these are all lymphocytes, white blood cells that are being produced. And look at the stem cell, hemocytoblast, the same one that made red blood cells. So hemocytoblasts can make red and white blood cells. Amoeboid movement. Your white blood cells do have the ability to move through your tissues. They can move through your tissues to a source of an infection to kill the bacteria at that site of infection. You can look at them and they all have a nucleus. All the white blood cells have a nucleus. They just look a little bit different. They have a nucleus, but they do not have hemoglobin. They don't have hemoglobin because their function is protection, part of your immune system. Their function is not to carry oxygen. That's the function of the red blood cells with their packets of hemoglobin. They carry out diapedesis, D-I-A-P-E-D-E-S-I-S. This is movement out of capillaries. So they can actually kind of move out of these capillaries and get into your tissues. And this has been happening your whole life, so it's not like something new is occurring here. Around 5,000 to 10,000 per cubic millimeter of blood, quite a bit fewer than the red blood cells, but they have a different function. Their function is your immune system to keep you free of bacteria and keep you healthy. They destroy bacteria, which are called pathogens, by phagocytosis. P-H-A-G-O-C-Y-T-O-S-I-S. It means cell eating. And by bacteriolysis, which means cell bursting or cell destroying. Well, cell will be bacteria. They have enzymes that will do that. Chemotaxis, let's fill your little blank there. Chemotaxis. Chemical substances which cause white blood cells to move toward or away from the source. The chemical substances are usually the waste materials that, the waste proteins that bacteria give off as their waste products, the products of cellular respiration. Those are not common to your body and they are foreign proteins. And your white blood cells will move toward the source of those proteins, which would be the bacteria, and kill them. Now, the lifespan of white blood cells, it says, is between 200 and 300 days. That's a pretty wide span there. And the reason for that range is the general health of the individual. If you're healthy, then the white blood cells are not going to be that active. They're going to be present, but they're not being active to actually like destroy bacteria or something, so you're not sick. So they can last quite a long time. If you're sick, their lifespan is going to last a shorter amount of time. Their activity is what determines their lifespan. If they're very active in fighting off diseases and stuff, you're sick a lot, then they're going to die quicker. If you're healthy, they tend to stay around a lot longer. They can be destroyed by microbes. Microbes can kill them too. The spleen and the liver filtering them out because they're not flexible. Bone marrow and lymph nodes can also filter them out. Now, types of these white blood cells. The first one is granular, Granular. And you look over here on the left side of this picture at the bottom, there are 3 of them that are granular leukocytes. These granules in there tend to stain, dark purple, something like that. They're packets of enzymes, compounds, and other components that are used to defend against pathogens, reduce inflammation, and destroy cells. So that's their ammunition is in those granules. And it's going to be somewhat different for each type of granular leukocyte because they have somewhat different functions. They originate or they're formed in myeloid tissue, red bone marrow. The hemocytoblast, which you can see there at the top, that was that stem cell. Immature cells formed from undifferentiated mesenchymal cells in red bone marrow. That's stem cells. It means stem cells. So let's look at some of our white blood cells. Okay, let's look at the first one, which will be neutrophils. Neutrophils. Neutrophils occupy about 65% of a count of 100 white blood cells. If you got a slide and you counted the field of views as you went down and then go over one field of view and come up and go over to the right one field of view and go down, when you got to 100 and you were only counting white blood cells, about 65 out of 100 would be neutrophils. They're the most abundant. They're phagocytic. They're called microphiles or macrophages, macrophage. It means little eater. They have a lifespan of about 10 phagocytoses. So that kind of determines their lifespan, how active they are. So they can phagocytose about 10 times or so. They release lysozyme, and lysozyme will pop bacteria. It ruptures them. So when these little granules in here can have packets of lysozyme that will pop bacteria. Now you see that this is called a lobed nucleus here. You have several lobes here. So it's granular and there are lobes. Okay, so you got to kind of get that. The background is going to be anywhere from clear to light violet with granules, and a lobed nucleus has lobes to it. Well, number 2 is an eosinophil. You see the eosinophil, it means the phil is lover of, eosin is a red stain, a red dye, lover of the red. So you look at this one, and there are several characteristics besides it's about a 3% presence out of a count of 100. These are associated with allergies, found in extremely large numbers when the body is infected with parasites, Parasites. Now this one stands out. It's very easy. None of these white blood cells look the same. Remember the first one, the neutrophils, had lobed nucleus, had granules. Well, look at this one. It's bilobed. It has 2 lobes. That's it. 2 lobes, like 2 little paddles. It has granules, and they're going to be somewhat reddish, pinkish to reddish granules and cytoplasm here. So this is the only one that has that pinkish to reddish cytoplasm, is going to be the eosinophil. The next granular leukocyte is going to be a basophil, lover of a basic stain. About 1% of a count of 100 will be these. And some books will say, 0.5 to 0. These are harder to find, but they have lots and lots and lots of granules on them. It's hard to see the nucleus most of the time inside of there. They liberate heparin. Heparin's an anticoagulant into the blood to prevent clotting. Well, it's going to be usually at an area that's wounded to prevent clotting so that more blood can arrive on scene, more white blood cells can arrive there to fight off the bacteria, and then a clot will form. They're in large numbers at the site of inflammation, and they are also involved in allergic reactions. So you see it's heavily granulated, so much so that you can barely even see the nucleus in there. Lots and lots of granules. So those 3 that we've looked at so far, the neutrophil, eosinophil, and basophil, all have granules, and they don't look alike. The neutrophil has that's a polymorphonucleate cell. It's got many lobes to it, a light violet to clear cytoplasm with granules. The eosinophil, 2 lobes. They're going to have 2 lobes. That's the special thing, 2 lobes. Kind of a reddish pinkish cytoplasm with granules. And then your basophil, just a whole lot of granules. It's hard to see the nucleus. On some of these cells, you won't even be able to see the nucleus. There's so many granules. So let's move over to the agranular. Agranular means without granules. So we have agranular produced in lymphoid tissue. And your 3 blanks there are the liver, the spleen, and bone marrow. Liver, spleen, and bone marrow is going to produce these cells. And the first one there, about 6% out of a count of 100, going to be a monocyte. Now this one, you got to kind of look at it. It's supposed to be shaped like a kidney bean, or shaped like a kidney. Okay, it's very easy to identify. Shaped like a kidney bean, the nucleus is. It's phagocytic. In fact, it's called a macrophage. M-A-C-R-O-P-H-A-G-E. That's what it says, become macrophage. It means big eater. They can phagocytose up to like 100 times. So they're much more powerful as far as phagocytosis goes than a neutrophil. More powerful phagocytes than neutrophils. That goes in the blank, neutrophils. The last one we have to look at is called a lymphocyte. Lymphocytes have a large round nucleus, and you can see a little bit of cytoplasm, and there's no granules. Okay, so lymphocytes, L-Y-M-P-H-O-C-Y-T-E-S, about 25% of a count of 100 are going to be lymphocytes. Play a large role in immunity, like T lymphocytes, B lymphocytes. Your T lymphocytes are what the HIV virus wipes out. So it's going to wipe out a severely powerful part of your immune system. Become phagocytic macrophages at the site of inflammation. So they can become macrophages also, and they're very important to have. Thrombocytes. Now you can see some thrombocytes here. See these little granules up here? It's like little specks of stain or dirt. Those are thrombocytes. They're platelets, have no nucleus. So here is a picture. There's your hemocytoblast there, your stem cell again, and look what it does. It makes a cell called a megakaryocyte that makes these platelets. So it says formed from megakaryocytes. which fragment while still in the bone marrow. So there it is, megakaryocyte, that goes in the blank. Around 300,000 per cubic millimeter of blood. That's a lot. 2 to 4 microns in diameter, or oval discs. You can see the little fragments there. And on the slide we saw just a second ago, this one, you can see some there. So if you see a blood slide, you see stuff like this around in the plasma, those are thrombocytes or platelets. They have about a 1-week lifespan, not very long. They're constantly being made, and to keep your ability to form blood clots. Contain thromboplastin to start the clotting mechanism. So they contain something called thromboplastin to start the clotting mechanism. Now moving back to the hematocrit again, let's look at the plasma. About 55% of whole blood is going to be plasma. This is the liquid part of blood, and usually it's going to be somewhat straw colored, which is kind of a light golden brown color, and it kind of varies depending on whether you're processing a lot of red blood cells or not. It's going to change the color of your plasma, and also medications that you're taking can change the color of your plasma too. It's the whole blood minus the cells. It's the plasma left behind. And what goes in that little percentage slot there is 55% of whole blood. The plasma is roughly 91% water. And I gave you some contents here. I listed the contents there on your notes. Other things you can find in the plasma. So there are a lot of electrolytes, some proteins are in there, vitamins are in there, hormones, enzymes, the kind of things are in here too. I have a blank. Serum, S-E-R-U-M, is plasma minus the clotting factors. That's what serum is. And sometimes when you give blood, they'll get your plasma and take the clotting factors out, and what's left over is called serum. An example of the fluid expressed from a clot. If you've ever gotten a scrape or something and you form a scab, you probably notice that there's a little droplet, a clear droplet on top. That is serum. All the clotting factors have been used with the red blood cells to form the fibrin needed for a clot and to trap red blood cells. What's left over is just the serum. Now you have a blank there. It says a foreign protein. Foreign proteins are called foreign antigens. They're foreign antigens. So antigen goes in there. And when we talk about your immune system, those are foreign antigens because you have natural antigens that are part of your own cell identity. Antibodies, special proteins produced by the body that inactivate antigens. So when you have a bacterial infection, the bacteria have the foreign antigens on the surface of those cells. And your white blood cell sees those as being foreign antigens and will, through a process we'll look at, produce antibodies that will bind to those bacteria and eventually shut down the receptor sites where that bacterium cannot communicate with the environment anymore, and it dies. So an antigen-antibody response. Okay. When an antigen enters the body and reacts with B lymphocytes, the B lymphocytes become plasma cells, and we'll look at that more in the lymphatic chapter. They become plasma cells, P-L-A-S-M-A. The second word is just cells, C-E-L-L-S, which are capable of producing antibodies which will attach to the antigen. The antibodies are antigen specific. They were made for those antigens on that foreign cell. So it's not one antigen for all, not one antibody for all antigens. It's pretty much one antibody, one antigen. They're made for each other. The plasma cell makes the antibodies that will bind to the surface antigens on those foreign cells, whether it's bacteria or, let's say, from a tissue transplant, like an organ transplant. People that have organ transplants have to have their immune system suppressed so that their immune system does not attack that foreign organ and kill it. That foreign organ was put in your body so that you can live. Now blood clotting pathways here. Blood clotting, let's see. A cut vessel or ruptured vessel contracts to reduce flow. Now, how does a blood vessel contract? Well, if you remember the profile of arteries and veins, they have smooth muscle around them. And when the smooth muscle contracts, it reduces the blood flow. So they'll immediately try to reduce the blood flow so you don't bleed very long, and it gives your clotting mechanisms more time to plug the hole or stop the leak. Platelets attempt to plug the opening in the vessel. Okay, now inside of blood vessels, the inside lining of the blood vessel is basically charged negative. Platelets are basically charged negative. Now, do same like charges attract each other? No, they repel each other. But a broken vessel has collagen fibers which are positive, and that means the platelets are going to be attracted to the collagen, and they'll bind to them and start a clotting mechanism to occur. So platelets attempt to plug the opening in the vessel. When they contact the collagen fibers, they become sticky and secrete ADP, which perpetuates the cycle. Well, it perpetuates the cycle to draw more platelets to the area, and then more platelets stick to the collagen, and it eventually plugs the hole. A clot forms, the growth of fibrous tissue into the clot to close the hole in the vessel permanently. That's going to be a little bit of scar tissue. It's going to be collagen. So the fibrin in the cells will make the plug to prevent the vessel from leaking, forms a clot, and then the repair mechanism goes on, and the repair is going to replace that clotting material, the cells and the fibrin threads, with collagen to make a new wall across that hole so it doesn't leak. Now, that's a similar method that happens if you get cut. Sometimes you scar, well, your cells repair the area and excess collagen is laid down in that area, and we call that a scar. Now here is something right here that we're going to talk about. Here are red blood cells, and the clotting mechanism has started the formation of these real thin little threads here called fibrin, and the fibrin makes a real woven net-like pattern or arrangement that the red blood cells get stuck in as they try to pass through the web of fibrin fibers. And enough of them get stuck because they can't pass through, and they form a plug, and that blocks the flow of blood out of that vessel. Okay, so we have coagulation, the process of clotting. We're not going to get real deep into this. Just know the basic on this. The first one is the extrinsic pathway caused from damaged tissue. It's on here to the right. Tissue, this is trauma to the tissue. An extract from the damaged tissue thromboplastin, is released into the blood. Tissue thromboplastin reacts with calcium, and you can see calcium is involved in blood clotting. So calcium is not just involved in strong bones and teeth. You knew that calcium is also involved in muscle contraction, and calcium is also involved in blood clotting. So tissue thromboplastin reacts with calcium ions to form extrinsic thromboplastin, which is going to be one of these down here. The names have changed. Extrinsic thromboplastin and several factors react to form prothrombin. Okay, here's prothrombin. Yeah, okay. Which reacts with calcium to form thrombin. There's thrombin. Thrombin reacts with calcium, there's calcium again, to catalyze the conversion of fibrinogen. Remember I told you fibrinogen, the ogen is an inactive precursor molecule. Fibrinogen into fibrin. And fibrin is those thin strands of fibrin, or what form that network that red blood cells get trapped in to prevent blood flow. So prothrombin reacts with calcium to catalyze the conversion of fibrinogen, F-I-B-R-I-N-O-G-E-N, to fibrin. Let's look at the intrinsic pathway. The blood itself is traumatized. And this is what I was talking about a minute ago where the blood vessel breaks. The endothelial lining and platelets are both negatively charged and repel each other. Therefore, they don't stick together. Platelets will stick to a ruptured lining. Why? Because the collagen is exposed. Underneath those simple squamous epithelial cells that form that endothelial lining is collagen. And collagen is positively charged. And the platelets are negative, collagen is positive, they attract each other. Platelets release coagulation factors to form intrinsic thromboplastin. Again, it's going to be down in here, even though the names have changed, but this is happening down here. Platelets release, intrinsic thromboplastin helps convert prothrombin to thrombin. Thrombin helps convert fibrinogen to fibrin. Okay, so one is tissue damage, one is the vessel damage. They both have the same end result, the production of fibrin, which helps to block the leakage of blood through a vessel or into the tissues. Now, clot retraction occurs within 30 minutes to close the wound. So back to here, you have all these fibrin threads here in the red blood cells. You have a scab form because of the clotting factors. And you probably noticed that when you form a scab, it feels like it puckers a little bit, kind of pulling. That's the clot retraction, to pull the tissue together. Occurs within 30 minutes to close the wound, and serum is expressed from the clot. That's that little droplet of fluid I told you would be at the top of your scab. Now I have some words here for you to look up, for you to find the definition of, okay? Fibrinolysis, not hard, plaque, thrombosis, embolus, and embolism. You need to look those words up. You may see them on your next test.